Kia ora koutou katoa, everyone. Welcome to our second Women in Science event for 2022. I'm the Head of Research at the Neurological Foundation and I'll be hosting our discussion tonight. We'll be keeping some time at the end for questions from the audience. So if there's anything specific you'd like to ask our panelists, please type your questions in the Q&A box. So our Women in Science events give us an opportunity to highlight the talented women working in the field of neuroscience in New Zealand. And tonight we're very privileged to have 4 of them join us. Bronwen is a professor in pharmacology and head of the Neural Reprogramming and Repair Lab at the Centre for Brain Research at the University of Auckland. Bronwen has supervised a large number of PhD students that have had Neurological Foundation PhD scholarships, and she's joined by 3 of them tonight. So Amy received one of our scholarships in 2013, Nicole in 2016, and Brooke was awarded one just last year. So I'll let each of them tell you a bit about themselves and how long they've been involved in research, what they're currently researching. So let's start with you, Bronwen.
Great, thanks, Sarah. As Sarah said, I head up the Neural Reprogramming and Repair Lab at the Centre for Brain Research. So I have been at Auckland University since 2000, where I joined as a lecturer after spending 3 years doing a postdoc position at Northwestern University in America. And while I was at Northwestern, I was doing research into gene therapy for the treatment of Parkinson's disease. And during that time, there was one of the really serendipitous findings that came about in research, which demonstrated that the adult human brain has resident stem cells that were able to make new brain cells. So I came back to University of Auckland, started up my lab with the idea of trying to repair the brain with neurological disease or neurological injury using those resident stem cells. And that was about a decade of research, which led us a long way down a pathway of really interesting discoveries about the ability for the brain to actually repair itself. And then in 2007, the Japanese researcher Senya Yamanaka demonstrated for the first time that you could do this process, which is known as cell reprogramming. And so that's when you take, in this case, skin cells from a patient, and you can insert some genes, or pieces of DNA that tell the skin cell to turn all the way back and become an embryonic-like stem cell. So embryonic stem cells, which are the basic building blocks that basically builds the human body. And what he showed that you were able to do is actually take these mature skin cells and turn them back into an embryonic stem cell fate, which then meant that you could actually go forward and make specific tissues, and for our case, brain stem cells. And that was huge. And he was awarded the Nobel Prize in 2013 for this discovery. And so that really caught my attention, and I thought this was really exciting to be able to actually generate live human brain cells that we could actually study. Except we didn't want to go all the way back to an embryonic stem cell state. So what we developed in the lab in 2012 and demonstrated for the first time was the ability to take skin cells, put in 2 genes, which are the first genes that are involved in forming the neural tube in the when the fetus is developing, and actually specifically make brain stem cells. So we didn't go back to an embryonic state, we just went from a skin cell to a brain stem cell. And from there we could make various different types of cells you find in the brain, such as neurons and astrocytes and oligodendrocytes. And so we've been working on that strategy in my group for the last 10 years, basically building it up to the point now that we can make very specific brain cells to cure specific diseases, such as GABAergic neurons for the treatment of Huntington's disease, or dopamine neurons for the treatment of Parkinson's disease, just for an example. And we use this technology to try and better understand the mechanisms by which neurological diseases occur. So we can actually study cells that we've generated from a patient with those actual diseases. We can actually have a look at the pathways, the disease pathway, and hopefully identify targets that we could use for drug development. But really excitingly as well, we can also look at transplanting some of these brain stem cells for the treatment of diseases such as Huntington's disease and Parkinson's disease. And we've got a very big program at the moment where we are looking at transplanting cells that are specifically going to turn into medium spiny neurons that are lost in Huntington's disease for the treatment of Huntington's, and which we're hopefully going to be starting to take into the clinic in the next several years. So it's probably just touching a little bit, but basically it's a broad scope of what we do. And Amy, Brooke and Nicole can sort of talk to you a bit more about the details of their individual projects.
Amy, how about we go to you next?
Thanks, Sarah. So I'm a postdoc in Bronwens group. I completed my PhD in 2018 under the supervision of Bronwen, and I basically stuck around because I loved the research and decided that I wanted to do more of it. So I'm currently funded by the Catwalk Spinal Cord Injury Research Trust, and my main focus through this funding is to use the cell reprogramming technology that Bronwen's just described to generate a different type of cell that is present in the brain and spinal cord called oligodendrocytes, and see how we can use them as a treatment for spinal cord injury. We've also got applications in the pipeline for this for multiple sclerosis. And through co-supervision of some of the other students in the lab, I get to dabble in lots of the other projects. So that's great fun.
Nicole, how about you?
Thanks, Sarah. So I'm a final year PhD student, and I've been with the Connor Lab since 2016 when I started my master's project. And at that time, I was looking at direct reprogramming approaches to studying a neurodevelopmental disorder called Fragile X syndrome as a proof of principle study at the time, and really enjoyed that work. So my current research is actually an extension of that work and focuses on establishing a human cell platform for modeling Fragile X using our novel direct reprogramming approach that Bronwen described, basically deriving human neurons from patient skin cells. And the idea is to use a suite of molecular and genomics techniques to identify what's going awry in these cells and throughout their development, and hopefully identify possible therapeutic targets. That's the plan.
And Brooke, tell us about your PhD.
Hi, so I'm Brooke. I'm the newbie to the Connor Lab. So I'm just about to hit my 1 year of being in the lab. So I'm currently working on my my master's project, and that's working closely with Amy on the oligodendrocytes that we generate and looking at their application for spinal cord injury. And then I was recently awarded a Neurological Foundation PhD scholarship that I'll be hopefully starting in end of this year, throughout the year. And we're going to be using the same cells, so oligodendrocytes, but looking at their use for multiple sclerosis. So very excited, and yeah, lots to look forward to.
So I think we're going to come back to you again, Brooke, because the next question is kind of for students that may be watching and thinking, how do you get to this place in neuroscience? What led you to be able to do a PhD in such an exciting lab where all of these neurological conditions are being looked at?
Yeah, well, it's definitely a very exciting field, and I do feel very lucky to be kind of have found myself in this spot here. But I know that for me, leaving school, I loved science. It was the only thing that I knew I wanted to do. But at that point, I just still didn't know quite how far the scope of science was. And so for me personally, as I was leaving school, my granddad was diagnosed with Parkinson's disease. And so at the time I was sitting there and I'm like, what is this brain and what is this disease doing? And so for me, that led to a lot of personal curiosity, just to question what's going on, what is this disease and what's being done about it. And so when I started my Bachelor of Science, I was majoring in pharmacology. I was doing various just generic science papers to begin with. And I just noticed that my ears would perk up every time the brain was mentioned, and, you know, specifically Parkinson's disease as well. And that kind of led me to continuing to take on these neuroscience papers. And through that, I met Bronwen, and, you know, she mentioned this, like she mentioned the cell reprogramming technology. And I just was like, oh my goodness, this is magic. It's crazy. And then it was even crazier when she told me that her lab had, you know, discovered this easier way to do it. And I just thought it was so amazing. And so, yeah, so I had to ask her a little if she had any spots available, and just, you know, being here and hearing of all the applications that this technology has is just crazy. But I think in terms of getting into neuroscience, it's just about really following what kind of interests you. And for me, like, I had that personal connection to neuroscience and the brain, and so just that curiosity, excuse me, is just what led me there, really. Yeah.
And what about you, Nicole? What's your research journey looked like? Have you taken the standard route or a roundabout route?
I definitely took the long way around to a career in neuroscience. I collected a few degrees in the humanities before I decided to retrain from scratch through a biomedical science degree, initially with a view towards medicine, before I basically decided to focus on more sort of upstream approaches to improving health. So I went from an honours to a master's in art history, and then to another master's in literature, trying to basically figure out where I wanted to be, what I wanted to do with my life, before I realised that I wanted to ask the question about what makes us kind of in a different way. And during my second master's, I lived with a friend who lost his battle to cancer, and it really made me kind of rethink where I could sort of best be placed to, you know, help people in a more direct and tangible way. And neuroscience in my mind was just a different way of asking the same questions I was asking in the creative sector about what makes us fundamentally who we are. So similar motivation, different strategies. And, you know, during my biomedical degree, I had in undergrad, you know, been exposed to Sir Richard Fall, got really inspired there. And when I stumbled across Bronwen through pharmacology papers and cell reprogramming, it sort of blew my mind, and here I am. So.
That's great. How about you, Amy? What's your journey look like?
I sort of don't know what I'm doing from one year to the next. So I finished school and decided to go to university. Wanted to study maths, so that's what I did, and then paired it with something that I didn't really know anything about called pharmacology. And then, similar to Brooke, along that journey, had some brain related lectures, and just thought that was interesting. Finished my degree, decided to do an honours year. Bronwen had also given me a number of lectures in my third year, and although it was not at all about cell reprogramming, I looked her up and found out about this magic technology. So she took me on for an honours project, which I really enjoyed and loved. So then stayed on to do a PhD, and then have never left. And here we are.
So you guys are reasonably early on in your careers, and Bronwen has sort of achieved the peak that people go for, is to get a professorship at the university. So what has your journey been, Bronwen, getting a permanent position as an academic and being able to grow your own lab? What's that been like?
So I think serendipitous is the word that I would use, which probably isn't what people want to hear in terms of planning careers. I also went to university with the idea of actually doing exercise science. So I used to row competitively, and I was very much interested in actually how to make sports performance better. And so that's what I thought I was going to do. And I took some psychology papers with the idea of, you know, sort of sports psychology. And that's really where I sort of found my passion. And this, you know, like everyone's just said, you know, the brain was just absolutely fascinating. Picked up pharmacology because I had 2 papers that I had to take to finish my degree, and I was flicking through the calendar and I thought, oh yeah, they look quite interesting, about drugs, and then just completely found my niche. But, you know, finishing up a PhD and going overseas, and I didn't ever really plan any of it. So the exercise science went out the window as soon as I decided that drugs in the brain were what I was really interested in. And most of my friends went on to med school. And, like Nicole said, I had this, I suppose, realization that I felt that I could help people better through research than I could if I was in the clinic. And so I'm always very mindful that I want my research to potentially have an outcome for patients, even if it's not necessarily our group that produces this, but it's helping along, it's helping with the building blocks of a potential therapy. And really all the way along in my career, the one mantra that I've always stuck to is to never say no to anything. So when I was finishing up my PhD, I went overseas to have a look for postdoctoral positions. Of course, I hate to admit that this was before emails were really, in vogue, and the internet wasn't really around. And so, I was writing letters to these people and waiting weeks for replies if I ever got them. And I had a couple of really good labs to go to. And so I was going over for a conference, and I added that in. And I just happened upon the lab of Marty Bone, which is where I ended up doing my postdoctoral position. And she was doing this technology called gene therapy, which at the time was just really quite revolutionary, and Parkinson's disease. And it just stuck. And it was sort of like, again, I think it's relationships that you build. You can't work for people if you don't have a good relationship with them. Even if that's a mentor and a student relationship, it has to be good. And I just instantly went, yep, this is where I want to go. And she was actually up in Rochester, New York, and she emailed me because email was starting to come in, but she emailed me and she said, I'm actually moving to Northwestern in Chicago. Will that change your mind? And I went, no, don't really care where I'm going. I just want to come and work for you. And so at the end of that postdoctoral period, I actually got what's now the equivalent of a Rutherford fellowship to come back and to work with Russell Snell and Richard Fall doing the transgenic Huntington sheep model. And at the time, Debbie Young, who's also well known to people in the neurological field, said to me, oh, there's a position open in pharmacology, you should apply for it. And I went, oh yeah, whatever, and chucked my CV in. And I mean, I can say this now. And chucked my CV in and went off on a 3-month trip around Europe. And in the middle of that trip, got invited to have an interview for the position. And so I actually interviewed at midnight in France and got the job. And I suddenly went, oh, okay, I've got a lectureship position. And it wasn't something that I'd planned. It was just like, oh yeah, okay, I'll put that in because I'll apply for that. Because if I don't get it, then that's okay. But if I don't apply, I won't get it. And so I then ended up coming back to Auckland and basically presented with this completely empty lab with absolutely nothing in it and no money and a course to teach, and told, off you go. And it has to be, I think, one of the biggest moments of building your personality and yourself as a researcher is just being given, no one's holding your hand anymore, and here you go. And that was a tough 6 months. And then I got my first project grant from the Neurological Foundation, which then helped me buy some stuff to put in the lab, and got some students, and then it just kept on going. And as I said, it was never anything that I really planned, except I always go and apply, and I always take the opportunity to apply for things, whether it's grants, whether it's promotion, whatever it is, because you're not going to get it if you don't apply for it. Okay. And that's always been my my belief. And also, as I said, not saying no to anything, unless he's like, oh my gosh, I shouldn't do this. But, if it feels good to you in your gut, you think, yeah, this is a good fit, I'm going to do this. And you go and do it. Because I think planning your career out can sometimes actually stifle the direction that, you just end up going down and towards, and yeah, it's been a great journey so far.
It's really interesting that your first real mentor overseas was another female researcher as well. And I've noticed that neuroscience seems to be quite dominated by women. Do you think this is true? Why?
I don't think it used to be. It is now. Well, it's seeming to be now. And maybe that, at the Centre for Brain Research, it definitely feels that way. I know that my mentor had a hard road as a woman. She was one of the few women in her faculty. She fought for everything she got, and you could tell that by the type of person that she was. So she was a fantastic mentor, but she knew how to fight for and she wasn't going to be, talked over or ignored. She was also there, equal partnership with the men in the room. In America, it definitely wasn't a woman dominated, it was definitely male dominated still. And I like to think that it's also a sign of the state that our society is in New Zealand, that I think that we have a really great society that just says, women can do anything. We live in a country that actually believes that and lets us do that. And neuroscience, for some reason, really attracts young women into it. So I think definitely we have a lot of strength coming up in terms of women in the field, and I think that's just really great to see.
What about at your level, Amy? I know there's a lot of fantastic colleagues that you have that are women. Is that something you've noticed?
Yeah, I think at my level, there's still a mixed bag, but there's definitely quite a few women around at the CBR. Yeah, I don't really know why it is. I mean, our lab group, for some reason, seems to have been a fairly female-dominated lab group as well. I mean, there's been some years where there are literally only women, and then a couple of young fellas come along. So yeah, I don't know what it is, but it definitely seems to be that way.
Nicole, you have been through other faculties as well on your journey to neuroscience. Is where you are now different in terms of the number of women?
I was surprised at how well represented women are in the CBR. They're obviously, I'd say they're fairly well represented in the humanities. I think that's pretty well established, depending on which department you're in. But yeah, I was pleasantly surprised. I'm not exactly sure about the percentages, in terms of how many there are of us, but let's just say, for argument's sake, we made up, say, 60% of the neuroscience workforce, for example. That still doesn't really tell you anything sort of about the distribution of women within that hierarchy. So, like 50%, for example, doesn't reflect the distribution of power, to be controversial. Like, power is the ability to exercise choice and be in positions where you can make meaningful influence. And so we know that there's an extremely high attrition rate of women from the workforce before 10 years post PhD, certainly overseas. I'm not so sure about New Zealand. And therefore they're underrepresented at higher levels of academia in all areas of research. And the important question, I guess, is why is that? So for me, there's a lot of PhD students who are female, that's well represented. But the further up the sort of hierarchy you go, I guess it's something worth looking at, and how well we're distributed across that sort of strata.
Yeah, that's a really good point. And one of the things I sort of think about is all of us know so many people with neurological conditions, and as Brooke mentioned, often we get led into the field because of a family member or someone close to us that has a condition, and we just have this desire to help them. Brooke, have you noticed that with other students that you talk to? Are they getting into the field because they have a personal connection?
Often that's the case. I've had a few friends that definitely have some kind of personal connection, which I think it's a great motivator, because it's one of those things when times get tough and it's nice to have a personal reminder as to what it's all for. But yeah, no, I think not everyone, but I think as people kind of illustrate their research more, and ordinary folk kind of learn about all of these different conditions, more and more people are kind of drawn into it. And I think that might be also, as women progress in society, and as society, we learn more and more through science, we kind of allow ourselves to be interested by various things. And I think that might be what push more women into science, or just, more people into science, perhaps. Yeah.
Yeah, that's really interesting. And obviously we can see that Bronwen has mentored a lot of young women through, many are doing PhDs in her lab, have done PhDs and gone on to get positions. And that's not the only engagement that Bronwen has with the younger generation. She's spearheaded a program called Being Brainy that many of you may have heard of. And actually, back when I was working as a neuroscientist, I had the privilege of working with Bronwen and going into primary and intermediate schools and helping to develop this curriculum that has actually been handed over to teachers that they can use themselves in schools. So I was wondering, Bronwen, if you want to tell us a little bit about that and how you think education and engagement is important to the future of neuroscience in New Zealand?
Yeah, sure. So yes, it seems a while ago now, Sarah, doesn't it? Being Brainy was sort of initiated around about 2014, and it came about because myself and some other colleagues in the department were quite often being asked by friends who were either teachers or friends who had told their teacher that they knew a neuroscientist to go into schools and to talk to kids about the brain, particularly when they started off at the beginning of the year and they were doing how do you learn and stuff like that. And I was sort of reflecting one day when I'd been to one of these talks, and gone in and we have the unique privilege of being able to actually take in plastinated human brains for the kids to see, and really unique experience for them. I always say to them, go home and tell mum and dad what you saw today at school when you're having dinner, because there's not too many kids that can actually say, oh, I held a brain today, so I sort of thought, this has actually got so much more scope than just, a few of us scattered around, popping in, doing this little show and tell and leaving. And so I got together a group of early career researchers and talked with them about putting together a program which has 8 weeks. It's an 8-week inquiry project for primary intermediate schools, and each week has so the first one is about the anatomy of the brain, the second one is about how brain cells communicate, and there's one on vision, there's one on hearing, and each early career researcher obviously was specialized in one of those certain areas, and they went away and they developed sort of like a lecture or a teaching program that would last about an hour and a half. And what I said to them at the time was that you need to take this into the schools, and it needs to be very hands-on. Okay, so kids don't want to just sit there and listen, they want to be doing stuff. So you need to have some hands-on activities, but they also have to be activities that teachers can actually do. So you can't say to them you need to have a microscope, because that's just not going to happen. And these early career researchers were just fantastic. They went away and they came back and they had all these really great little experiments for the kids to do, worksheets. And we trialled it out in several schools. We got really great responses from the teachers and from the students. And so from then onward, we developed it into a website, which got launched in 2017. And it's free. So schools you have to register just so we can keep track of what schools are using it just purely for an administrative purpose. But schools can go in there, and the programs, they range from right from year 1 all the way through to year 8. And schools can go in there and they can actually download the teaching plan for the teachers, the slides and all the information, all the worksheets. And that allows the teachers to then teach confidently about the brain. And I also say to them, you know, look, you can pick and choose. You can take this one, this topic and this topic, or you can merge it in. And we had one school who actually got really excited about it, and they had, the whole school did the program, and they did art, English, maths, everything around the project. They had sports activities, they had the Brain Olympics. You know, it was just really, really great to see the school and the kids get really involved. And so we also provide free professional development for teachers. But what it does is it allows, so I would always say to the teachers, I'm not looking to generate the next wave of neuroscientists. What I'm wanting to actually show to children, particularly at that primary school age, where it's really important that they get excited about science, is that, you know, what we're showing them is actually science. This is science. You're doing an experiment. This is what an experiment is like, okay? And that then gets them excited, but actually science is really cool. It's really fun. You know, it's not boring, because statistics show us that if we don't grab them by the time they leave primary school, they drop out of science. They're not interested. And so obviously, you know, being the brain being so fascinating, and everybody actually has a brain, so they can all relate, you know, and a lot of these kids as well too might say, oh, you know, my grandma has this. So it's very relatable, and it's relatable science, and it's fascinating because they're learning about themselves, and they can go home and tell mum and dad exciting facts. And we've just found it's been really well taken up. We've got over 450 schools that have registered and that do the program. And I just think it's just really great to actually get kids interacting and learning about their brain, because the last one we do about is what happens if I damage my brain. So they're also learning about brain health and the importance of actually looking after their brain, eating well, wearing a bike helmet, seatbelt, all those things. And it's been a really great and exciting program to work with, and actually Amy and Nicole have often come along and help out, and we've been to some pretty cool places. Last year we went down to Gisborne. I went to several schools down in Gisborne, which was great, because those schools just, you know, don't get the opportunity to have that type of interaction with scientists. So I think it's a bit broader than just neuroscience. You know, obviously we're presenting neuroscience, but it's actually science that we're showing them. And actually what I think is really exciting is that generally the group of people that go myself in that group, of course, as well. So, you know, they're also seeing that the scientists that they're seeing are not, dare I say it, old white-haired men. They're seeing young women or young men going down there, and we're scientists, you know, and they're like, oh, you don't look any different to my dad or my mum or my brother, or, you know. So it's making us look less scary, you know, as well, which is always good.
Amy, you've had a foundation grant as well as the Catwalk Trust, and both organisations are very focused on engagement. How have you found that? Do you find people are really surprised that you're a neuroscientist?
Yes. One of the first sort of meet a scientist visit that I went on through the Brainy program that Bronwen just talked about, I went down to the school in Christchurch, and I'd been emailing the teacher, and we'd organised everything. And then when I arrived, she looked at me and I said, "Hi, I'm Amy." And she said, "Oh, you're the scientist?" So I said, "Yes." And she said, "You're really young." So I said, "Thank you." And she said, "Well, I was a little bit surprised that you were a woman, but I could tell by your name. But yes, I am quite surprised as to how young you are." And I think a lot of the kids in the school thought the same thing. And there was actually some of the young girls who sat at the front, and I remember they were sort of burning to ask me some questions, and they did say exactly what Bronwen said. You don't wear glasses, you don't have crazy hair, you just kind of look like average Joe or average Beth. So that's quite surprising to us, and I think it was sort of refreshing for them that you can be a scientist and you can be a fairly good one, and you can also actually lead a fairly normal looking life.
Yeah, that's really fantastic. I'm just going to change tack a little bit because we're having such a great time talking. The time is marching on, and I really want to touch on the innovative and exciting aspects of your research because it's just kind of mind-blowing, really. Nicole, can I come to you next with this question? Just some of the most exciting and innovating aspects of your research that you've found so far.
Yeah, I mean, generally speaking, we're doing research that, up until 10, 15 years ago was considered impossible. And, establishing radical new methods to model disease in a dish by, changing the actual fate of a terminally differentiated human cell and then using it to understand questions about neurogenesis and disease progression is in and of itself amazing, So yeah, I'm excited by sort of possible applications of our reprogramming technology and reprogramming in general, both for disease modeling, drug discovery, cell transplantation in the future, those sorts of things. Yeah, so I'm just genuinely excited by the techniques that we're using and just the huge potential that they have, really. And what's
So
this
Brooke,
space?
we definitely will be, actually. So Brooke, just starting out, what has been the most exciting thing that you've come across just so early on in your career? You're still finishing your master's.
I think, it's very nice and easy for me to sit in a lecture room and to have Bronwen standing in front of me and going, we do this. And then to have the opportunity to walk into a lab, to feed cells, to watch them grow, and then I put them under the microscope and it's actually worked. It's honest, it's magic. It honestly is. And so for me, that's the most exciting thing, is actually seeing it. And just the other day, I was showing Bronwen and Amy, I stained some cells for the first time so I could basically assess that they were expressing something that we wanted to. And for me, I'm sitting there under the microscope, my heart's racing. I've stained my first cells, and it was just so exciting. So to take a human skin cell and to turn it into a brain cell, that's mind-blowing, and to be a part of it is it's an honor. I love it. So yeah, it's just the most exciting thing for me. And you're right, I'm only starting out, so I'm really excited. Yeah.
So Bronwen, at the other end of the spectrum, when you've been doing this for a while now, do you still find it innovative and exciting, and what really still grabs you about your research?
Yeah, I mean, it still blows my mind, as Nicole and Brooke said. It's, as Nicole said, about 15 years ago, before Yamanaka even demonstrated reprogramming as possible, I would have said it was science fiction stuff, and then here we are, and we're doing this, and I will never forget the first time that we actually achieved it. So I had, we talked about, in lab meeting, we talked about what we wanted to do. And it was a little side project, sort of slightly crazy side project that, we wanted to do this reprogramming, but we didn't want to make them like embryonic stem cells. We wanted to go straight to brain stem cells. And I put a postdoc that we had in the lab at the time, and my research technician at the time called Erin, and I put them onto the project, and I sort of said, off you go, give it a go, let's see what happens. And I sort of expected about, I don't know, these things in science can take a year. You change things, you change other things, you change, everything gets changed. And about 4 months afterwards, that meeting, Erin knocked on my office door and said, we've done it. And I'm like, yeah, no, you haven't. And she's like, yeah, we have. And I'm like, okay, well, I'll go and have a look down the microscope and I will see if you have or not. And I'll never forget looking down the microscope the first time and actually saying, oh my goodness, you actually have done this. And it was a very sort of eureka moment. And I think that's what, it's a bit like Brooke explained. Even though I've been doing this for ages, I still see things for the first time, so it may be that the technology or the, what we're doing, but even things like, transplanting these cells into a brain, this is the first time that's been done. And, we can do that. They survive. They turn into the mature neurons they're meant to be. That's the first time that's ever been shown. That is exciting. And for me, it's the type of job that just is never boring. Yes, there's some daily boring stuff, but in general, when you look back over, a couple of months or even the year, and you think, gosh, actually, we've done all this, and this is all new, and it's all exciting. And I think sometimes we get a little complacent about actually what exciting it is. For us, it's like, oh yeah, we can do this, yeah, and it is a bit like, it is magic. I still feel like it's magic, taking skin cells and turning them into brain cells that actually are functioning properly, can actually go and repair things. That's pretty cool stuff. So I'm just continually inspired, but I'm also continually inspired by these guys and the other members of my lab that come in. They're just continually inspiring people as well too, because of their passion and the desire to actually get on board my train and go along and actually and do this stuff. And that's actually really great to actually have people going, wow, that is this is really cool. I want to be part of this group. That's really great.
I think we're going to move into questions from the audience now. So if people have questions, feel free to start putting them in. But I think that leads quite nicely into one of the questions that we have, which is, what do you see the future holding for people living now with Parkinson's, multiple sclerosis, Huntington's disease, spinal cord injury?
Do you want me to take that one?
I think Amy's taken herself off mute.
No, go on, go for it, Bro.
I would like to think that the future looks pretty exciting for these people and these patients. We can't promise the earth. We can only go by what we think we're capable of doing at the moment. But we believe that this is really powerful technology, and we would like to see that in the next decade that this technology is actually going to start making some difference to the well-being and the lives of these patients with these diseases. Potentially through transplantation of these cells, or even through using these cells to identify new drug treatments. So, both arms are going in the lab. And the beauty, which we haven't actually touched on, but the beauty of using these human brain cells for drug development is that we're actually testing and identifying drug targets on human, patient-derived cells. And we use, basically as many different patients as we can handle at one time, and what it allows us to do is sort of like do what I like to say is like a little mini clinical trial in a cell culture dish. And we're not relying on engineered cell lines, or we're not relying on rodent cells. Eventually some of these drugs will have to go into animals to test, how they get work through the body and stuff like that. But the actual key technology is actually being developed in humans. And what's really crucial is that we're actually establishing these technologies in patients with different genetic backgrounds. And that's one of the key problems, is that when you develop drugs in rodents that are all inbred and pretty genetically the same, and you go into the clinic, as any clinician will say to you, no one patient with Parkinson's disease looks the same. Their symptoms are different, their onset's different, the duration's different. So you need to be able to have, and of course they've all got different genetic backgrounds. So you need to have a drug that can overcome all of that and actually work. And that's why most of the drugs that have gone in for neurological disease have not succeeded, because they can't combat that. And we're hoping that with using these human cells, human patient-derived cells, that we can actually find a drug that, or identify a target in a drug that actually is working well enough that it overcomes all of that variability that you see in a clinic, in patients in a clinic.
Yeah, that sounds really exciting. Amy, did you have anything to add about where you see the technology going?
I was going to say that I think the future is really bright, and that we're all very excited about it, and hopefully through various opportunities where we can speak to the public, we can make the public very excited about it and give them hope as well.
So when you meet someone with a neurological condition, what kind of things do they ask you? What do people want to know? Because it will be the same sort of thing that our audience are wanting to know tonight.
They often want to know if there's a treatment available and how soon, which is fair enough. And I think it's very difficult for any of us to say how soon. But what I always like to say is that there are plenty of us working on different strategies for treatments, or at least being able to relieve some of the symptoms. So with all of us and all the young and up and coming people like Brooke who are coming through, because there are a lot of them now, from everyone working on it, we should be able to get there soon.
Do you have any kind of timeline or time frame that you think they'll be ready to start testing drugs on the cells, Bronwen?
Well, our transplantation work is the stuff that's going ahead the fastest. And as I said to you, the work we're doing looking at transplanting these reprogrammed brain cells for Huntington's disease, we're looking at patenting that potentially in the next 6 months. And then, then it's working forward and getting it ready to go into patients. Probably what would be the big crunch for it would be finding investors to fund such a clinical trial. But we think that basically within the next 5 to 10 years, that technology will be ready to go into clinical trial, and we're adamant that that is going to have a very strong arm in New Zealand, because we have an incredibly supportive neurological community here in New Zealand of all the different neurological disorders, and we would like to give back to that community and have them fully involved in clinical trials of this type, and not see it just go overseas.
That would be really fantastic. So we'll keep our eye on that. For the next question, I'm going to come to you, Nicole, because I know that you've been trying to finish off your PhD during COVID. And normally PhD students will be traveling overseas, presenting their research, getting feedback and all this kind of thing. So do you want to tell us a bit about how the pandemic has been affecting your research and the wider lab?
Yeah. Sorry. Yep. So yeah, COVID's absolutely impacted the entire research infrastructure, funding, reagent delivery and delays, inability to travel to conferences and engage in networking and career development opportunities that we would have had in the past. I mean, a lot of these have gone online, but it's really not quite the same. And there's sort of most professions, there's sort of an emotional toll that comes with navigating uncertainty that comes with COVID. And one of the key struggles I think most researchers have, and probably most people in general, is trying to get that work-life balance in a post-pandemic world where it's sort of the overlap between work and home, or working extra hours to compensate for time that we lost to lockdowns can really skew the balance. And I think it's really easy to sort of forego your own well-being to achieve an end goal that's really important to you. Yeah, and I think it's just the delays and the uncertainty. I'm supposed to be heading off to the US and that's all just been sort of put on hold until I've finished. And it's just sort of trying to be resilient and adaptable and roll with the punches and make the best of it, because we're all, this sounds very glib, but we are literally all in it together, right? So we're all experiencing the same sort of issues, really.
I'm really interested.
sorry, just to add into Nicole's comments there, I think, we hear a lot about the effects of businesses of COVID and all the rest of it, but there's actually been a really major effect on the research community, as Nicole said. We've been unable to go into the lab and to do research for the majority of all the lockdowns that we've had in Auckland. And not only that, but when the lockdowns occurred, we had to destroy and discard months of experimental work. And that's quite heartbreaking. I mean, there was in the August 2020 lockdown, Amy and I went in, we had that really quick turnaround. And both of us were almost in tears. We were dumping about $30,000 worth of research in the bin because we couldn't come in to look after it. And, there's been a huge toll on the group. And I think it's a huge testament to all the researchers in my group and researchers within the scientific community that we've all actually kept strong and just kept on plugging on and sat at home, as I still haven't been back into the office it's because I'm not allowed to since August. So I'm directing from afar, so it's been a completely different world the last 2 years. And that has affected research, but the research is, I think such a powerful bunch of people with such commitment that they just keep on going.
Do you want to tell us a little bit about how the university is prioritising who's going in?
Well, at the moment with the red traffic light system, it's good. When we was in level 4, obviously no one was allowed in. So the last lockdown for us, because level 4 in Auckland lasted for such a long time, basically the lab was just, all the research labs and the faculty were dormant. Level 3, they then prioritised based on deadlines. And so we got Nicole in because she had a PhD deadline. But I had 2 other PhD students who were in their first and second year, and they couldn't go back in until we got to whatever it was, level 3.4 or whatever it was, picnics with people outside. We had some people allowed in because they had pressing deadlines. Brooke was also allowed in. But then I had half my group in and half my group not in. And that was, that was good for those that could go in, and it was tough for those that couldn't. Yeah.
So the foundation's made a real point of helping people with, financially to finish their projects, which I know has been helpful for people. Would you say, though, that the research has been set back quite a bit, or do you think you're able to catch up that time and do the work that you're planning to do?
So the Neurological Foundation has been fantastic. We basically, we were informed that we could let you know what had gone on, what we needed, and we were basically given it. And it was incredible support, and we have to thank you for that. But we're never going to make up that time. We are basically pretty much a year behind schedule. And, I think if you have a look at the amount of time that Nicole's had to extend her PhD deadline, that sort of is an indicator not on Nicole's capability, but on the fact that she has been unable to go in and do the research. And there's no way of catching up that. We had some of the strictest lockdowns in the world, so researchers in the States and in Europe were still going in staggered lots, whereas we couldn't go in at all. So it is what it is. We were all in it together, and as we say, and I think as long as we've lost that time, you can't make it up. There's no point getting upset about it. It is frustrating at the time when it's happening, though, but the support of the Neurological Foundation was just fantastic, and that was great to know that you guys had our back, basically.
Yeah, we definitely wish you all a lot of luck with getting in the lab, staying in the lab, and being able to finish off your projects. I just want to ask one more question that's come through from the audience, which is quite interesting because it talks about just other therapeutic approaches. And I know that you're looking at one therapeutic approach, and sometimes people might benefit from multiple therapeutic approaches for different things. So Bronwen, have you worked with other groups that are doing different therapeutic approaches or anything like that in your career so far?
Not to the extent of like different types of therapies, but you know, I think because you know we're focused, there's only so many variables you can put onto an experiment, right? And we're very focused on what we do really well, and then what I think though, there isn't just one way of doing this in terms of, you know, it isn't like you say, for example, you transplant the cells and that's it, that's all you're going to need. You're then going to need, you know, you probably have some advantage through physiotherapy, occupational therapy, music therapy, anything at all that has demonstrated that it has some potential to enhance brain function, well-being. So it's not a standalone thing, and no therapy really is a standalone thing. It's just that at the moment we have to focus on one aspect of it. And then it would be really interesting, you know, later on down the line to actually have a look and see how we can actually even further enhance that with some types of therapies that are not involved in transplanting cells into the brain, for example. So, you know, that's always a really exciting and interesting concept to start thinking about. Yeah.
Do you think research will get to the point where what happens in the lab and then gets into the clinic will be combined with other therapies just to make the brain really receptive and healthy and able to really take on these new technologies?
Yeah, and I think we already know that. You know, we already know, for example, and not even just with the brain, but you know, you break your arm, for example, it's very simple concept, right? You break a bone, you put in a plaster to repair it, but then when it comes out, you do physical therapy or you know, and some exercises to strengthen it and stuff like that. So, you know, repairing anything is not just a single shot aspect. You're going to need to have some whole body function or interaction there to help as well.
Well, our time is up, but I feel like we could keep talking to the four of you all night and picking your brains about great ideas for neurological research. But we just want to say a huge thank you for taking the time to talk to our audience tonight, and just want to thank our audience for joining us as well to learn more about neurological conditions and the exciting research that is happening. And I also want to just say a huge thank you to those that support the foundation as well and enable this research to happen. Thank you, everyone, and good night.